explaining. They should be able to fix these things and get them on time. All right, so today's Friday, and I'll stop the lecture 5 minutes early, and you'll take your standard Friday quiz. This one will have questions from the nucleic acids chapter and from the cells chapter that we're on now. All right, let's see. Special announcement. So as you know, Rex has been very kindly just voluntarily videotaping the lectures and has begun to post them on YouTube. And he sent me the link address. As soon as I'm done teaching here, I'm going to go in and put it up on the course website. And so click on there, and you can see the videos he's been so kindly making. We used to have someone from the college, a paid person to videotape, but there was sort of a mix-up this semester and they weren't able to get it together. So Rex, thank you for stepping into those, filling those shoes. All right, and by the way, I think you're doing a better job than the old people we had. They never seemed to keep me in focus. Or who knows, maybe I look better out of focus. I had an aunt who had that laser surgery on her eyes, and so then her vision got better. She said, oh my God, I've got a bunch of wrinkles I never knew I had. So she wanted to go back to her old vision. All right, let's see. Let me recap some of the main concepts from last time because it's kind of a complicated thing. This was all about how cells express their genes. And when you say express a gene, it means make a protein from the gene, because that's really what the genes are. They are the cell's recipes for proteins. This is supposed to be a gene, part of a segment of one of the chromosomes that encodes a particular protein. And remember the way the system that the cell has for encoding proteins with the DNA. The DNA is a series of G's and A's and C's and T's, nucleotides, but the proteins are a chain of amino acids. Well, the way this works is every 3 nucleotides in the gene is called a codon. Think of that as kind of a word that stands for one of the amino acids. That's a good example. A GCG codon in the DNA stands for the amino acid alanine, and then ACG codon in the DNA stands for the amino acid threonine. And the only thing tricky about this is this codon table they have here is written in RNA instead of DNA, but that's fine. Just remember that U's and RNA are the same as T's in DNA. So the codon in DNA, CTT, stands for the amino acid leucine. All right, so this particular gene, ATG, would stand for the amino acid methionine, GA stands for the amino acid glutamic acid, CCE stands for the amino acid proline, and the TGA, which is UGA on this table here, is a stop codon, which says this is the end of the gene, no more amino acids in this protein, and that's how this gene encodes this particular protein. All right, now the genes are parts of chromosomes, and chromosomes are the nucleus. To get that genetic information out to the ribosome, which is the organelle that actually assembles the amino acids, a temporary disposable copy of the gene is made, or I should say of one strand of the gene is made, and that's called a messenger RNA because it sends the message, the codons out to the ribosome. And that step is called transcription, when the messenger RNA is made from the gene. And then notice the nucleus, the membrane around the DNA has holes in it to allow these messenger RNAs to exit the nucleus. Then they travel out, they go into the cytoplasm, they fit between the large and small units of the ribosome, and then, this is the details of the transcription process. Go back. There we go. And then the ribosome will then begin the process of translation, which is where the ribosome actually makes the protein from the codons in the messenger RNA. And this is just some details about the transcription process. The 2 strands of the DNA separate from each other. An enzyme called RNA polymerase binds to one of the strands and makes a RNA complementary to that strand. But if you've noticed, the RNA that was made, the messenger RNA, has the same sequence as the top strand of the DNA. So in essence, by making a strand complementary to the bottom strand, you get the same sequence as the top strand. And they call this the template strand. The strand that the RNA polymerase runs along is the template strand. And the strand of the DNA that the RNA polymerase doesn't run along is called the non-template strand. So in other words, the messenger RNA is the same sequence as the non-template strand. And then the messenger RNA, when it's completed, detaches from the DNA, exits the nucleus, goes into the ribosome, and now the ribosome starts the translation process, which we have right here. And the amino acids just sort of float around in the cytoplasm, and they come to the ribosome and get, ooh, this is a good review question. What's the name of the bond that links amino acids together? Peptide bond, yeah. Peptide bonds are formed between those amino acids that link them together, and that's how you get the protein. Yeah, I think I ended on, I showed this last time, but I ended on this thing called transfer RNAs. The cartoon that I showed you up to this point is not 100% complete. There's one more organelle that's involved, or a structure that's involved, and these are called transfer RNAs, or tRNAs for short. This is the tRNAs are RNA molecules. And to show that fact, I included some RNA nucleotides down at the bottom, but the whole thing's made out of RNA. And the tRNAs hold amino acids. They are the carriers of the amino acids. And so when I showed you those amino acids floating around in the cytoplasm, what I wasn't showing was that each amino acid is being carried around by one of these tRNA molecules. But maybe the most important part of the tRNA is this. At the bottom, they have 3 of their RNA nucleotides that sort of hang down. And that's called the anticodon because it is the complementary sequence to the codon for whatever amino acid the tRNA is holding. If we go back to the genetic code for a moment, the codon for the amino acid methionine is AUG, and so any tRNA that holds methionine will have the complementary sequence to AUG, which is UAC, as its anticodon hanging down there at the bottom. And that's important. We're going to need the tRNAs to be carrying the anticodons for whatever amino acid they're carrying, because that's going to be necessary for them to match the codon on the mRNA up with the proper amino acid by complementary base pairing. So here's the full and complete picture. Each amino acid in the cytoplasm is held by one of these transfer RNAs, these tRNAs, and when the messenger RNA comes out of the nucleus and clicks into the ribosome, the tRNAs shuttle their amino acids in, like this. Let's see, there they are. But notice the part here that what allows each amino acid to be brought in the proper sequence is the anticodon on the tRNA matching up with the codon on the messenger RNA. So that forces the first amino acid to be methionine, and the next amino acid to be glutamine, and the next amino acid to be proline. I think when I was talking in the nucleic acids chapter about complementary sequence, nucleic acids will always magically find each other and bond to each other. And so that property is being exploited here to match up the proper amino acid with the proper codon in the messenger RNA. Sound good? All right. And there we go. And now we've completed the protein. The tRNAs, now that they've done their job, detach themselves from the amino acids that they were carrying. what will they do? Well, they'll go back and eventually link up with a new amino acid so they can be ready for the next protein that's going to be made. Yes?
Each tRNA is specific for a certain amino acid?
Correct. Right. Each tRNA will only be matched up with the amino acid that it has the anticodon for.
What carries the mRNA from the nucleus?
What brings the messenger RNA out of the nucleus to the ribosome? I think it's just random diffusion. That there's no train track that it goes along. It just drifts out in the cytoplasm and eventually just kind of randomly bumps into a ribosome.
There's no structure in which that thing is going to
No.
And the ribosome is a structured organelle within the cell.
Yes, yeah, it's partially made out of protein and partially made out of RNA. Yeah, good questions. All right, so I think this next slide kind of sums up all of this. The purple thing is the nucleus. All right, so inside the nucleus, the messenger RNA, which is the light blue thing there, gets made from the DNA, which is the purple-looking thing. And then the messenger RNA exits the nucleus, which is why the nucleus has those little holes in it, goes out in the cytoplasm. The ribosome, small and large subunits, clamp down on the messenger RNA. The transfer RNAs, which look like little seals balanced from a circus balancing a ball in their nose, bring in the amino acids to the ribosome, and then using their anticodons, they pair up with the codons on the messenger RNA, and that brings the right amino acids in the right order, and you get the protein formed. All right, good. Let me switch to a new computer cartoon thing here. All right. At the beginning of this chapter on cells, I told you that there are two different types of cells on Earth. There are prokaryotic cells and eukaryotic cells. And what was the defining difference between the two? Anybody recall?
The eukaryotic were single cell?
No.